So, any doubts in yesterday's class? If you have any doubts in yesterday's class, first I'll clarify them and I'll go for the next topic.
No, ma'am, no.
Okay. So our today's topic is, as I said you yesterday, our today's topic will be clinical trial protocol. What is clinical trial protocol? What the things will present in clinical trial protocol, and what is the importance of clinical trial protocol? Okay.
Okay, ma'am.
Give me 2 minutes. So first we should learn what is the protocol. Protocol, as I said you, it is a document where it will explain us about the study completely, how to handle the study, what is the thing we should do, what are the processes we have to follow when we are conducting the study. Those all things will we can know from the protocol itself. That's the reason here you can see it is a document that describes the objectives, what is the main objective of our study, what is the design of the study, and what is the methodology, and whatever the results we get from the study, how we'll calculate them using statistical consideration, and complete organization of the clinical trial, how we are going to do the clinical trial, everything will be mentioned in the protocol. So every clinical study, every clinical trial, whenever we are conducting a clinical trial, it begins with the protocol. Without protocol, we cannot do the clinical trial, because in protocol only we have everything, the design, methodology, the objective of the study, everything. So without that one, we cannot conduct the clinical trial. That's the reason every clinical investigation will begins with a protocol only. And it contains the main things. What it will contain is background for the study. Why we are doing the study? What is the background? For example, now I am doing for example, your most known product, I'll tell, ketorolac, mainly for dental pain, they'll be prescribing ketorolac drug.
Yeah.
So in protocol, they'll mention the background first. So why we are doing this study? Don't we have any studies on this product previously? So in that background, they'll mention previously there are studies, but now ketorolac is present in tablet form. Now we are doing in gel form or injection form or some other formulations. Those all background they will mention. Next, rationale. In rationale, the importance of the drug. So why only ketorolac? Why not other drugs? So that is nothing but the rationale for the study. That rationale will mention in the rationale section. Next, objective. I think you must have done your project in BDS, and at the time you must have mentioned what is your objective, what is the main objective of your project, and to
Yeah.
achieve that objective, what are the methods you are going to do, and after doing those all methods, whichever the results you obtain, for those results you must have done some statistical like statistical analysis also, like keeping graphs, from those graphs attaining some values, and at last showing the result of the study, like that we'll be
Yeah.
doing. Same thing we'll do here also, but those all things generally in our projects, academic projects, what we will do after doing everything, we'll mention those things in a thesis,
Yeah.
right? But here, before doing the project, first we will write a protocol clearly what we are going to do, and then only we'll do that process.
Okay.
Okay, so that is the protocol. That is the difference between the protocol and the thesis. In thesis, we'll mention everything what we have done, the methodology, the results we obtain. But here in protocol, what we are going to do, that one we will write. And again, after getting the results, we'll have a clinical study report. We'll call it as CSR. In that one, we'll mention the results.
Okay.
Okay, so what is the general information that will present in the protocol? The first one is protocol title. Protocol title is nothing but the study title which we are going to do. Next is protocol identifying number. For every protocol, there will be a specific number, because for all the protocol, if you are keeping, if you are don't keeping a number, there will be a confusion to know which protocol is for which study. That's the reason for each and every protocol, there will be some unique number which will help us to know about that study. And date when it was finalized, the finalized date we will mention. Any amendment should also be the amendment numbers and dates. As I said, if any changes are done in the current protocol, then in the amended protocol we'll keep the version. If the previous is version 1, and in this amended protocol we'll keep version 2, and when the version 2 date was effective, that one we'll mention. Next, name and address of the sponsor. Who is the sponsor for that study, and what is their name, their address, and everything. Next, monitor. Monitor is nothing but CRO. Okay. So
Okay.
CRO details also we'll mention. Next, name and title of the person authorized to sign protocol and protocol amendments for the sponsor. So from sponsor side, who was the main person to approve the protocol, and their signature.
Okay.
Next, name, title, address, and telephone number of the sponsor's medical expert or dentist, whoever appropriate for the trial. So here, from the sponsor side and from the CRO side, we'll have one medical expert. So their main their main responsibility is, whenever we are conducting one new study, that medical expert will check everything, whether we can do this trial, or whether it is feasible to do with this product, is this product is safe, because the only medical people will know about those products and how safe it is for the patients, right? And even when any adverse effects or adverse reactions are happening to the patients, they will take decisions whether to stop the clinical trial, or we can conduct the clinical trial by taking some precautions. These all kind of some precautions will be given by the medical expert. And if we are doing any dental kind of studies, dental doctors, they only can give us that decisions. That's the reason
Okay.
even we should have one medical expert from CRO side and one medical expert from the sponsor side to take decisions in these kind of things. Okay.
Okay.
Next, name and title of the investigator who is responsible for conducting the trial, their address and telephone number. Okay, so these all come under the general information sections. Section will give some general information to the person who is reading the protocol. So there we are letting them know about who is the sponsor, who is the CRO person handling the study, who is the investigator. Investigator is nothing but the doctor who is conducting the study, their address, their telephone numbers. And in studies, even we'll have some lab tests also. So for conducting those lab tests, which is the laboratory involving. So for some studies, what we will do is, even though the studies are conducted in different sites, for example, I am doing one pan India study. My sites, I have in Hyderabad, I have one site, in Bangalore, I have one site, in Chennai, I have one site, in Nagpur, in Pune, like that in overall India, in Kolkata, in different zones, in different hospitals, and in different cities, I am doing my trial. But I want to keep one laboratory only, in order to maintain some originality of the values, because from lab to lab the ranges will vary, right? For the
Yeah.
local labs the ranges will be different, and for the central labs the ranges will be different. That's the reason, mainly in most of the big studies they will involve central lab only, because central lab, what will happen, they'll have their own branches in different cities,
Yeah.
and they'll have same ranges only, they'll maintain same lab ranges only in wherever the location they are, they'll have their own standards, they'll have same methodologies, they'll have the same value ranges. That's the reason in most of the studies we'll take central labs only. Okay. So
Okay.
the central lab details also we have to mention in the protocol. Next, that is about the general information. Next, as I said, background also we'll mention in the protocol. So that what Sorry, I'm suffering
Okay.
cold today. Okay. So in background information, what kind of information we have to give in the protocol? In that one, name and description of the investigational product. The name of the investigational product and its description. Its description is nothing but its physical, its physical properties, its chemical properties. Those all things we'll give in the description. Next, summary obtained from non-clinical studies that potentially have clinical significance related to clinical trials. So, previously, when we are like before conducting the trial in human beings, first we'll conduct the trial in animals, right?
Yeah.
So, that data, whichever we obtain from the animals, that data we'll call as non-clinical data. Those studies we'll call as non-clinical studies. So from that studies, whichever the summary is nothing but overall the results which we obtain from the animal data. Those results will summarize and we'll write in this second one. Summary obtained from the non-clinical studies. What data we obtained from the non-clinical studies for this investigational product. Those all will mention in the background information. This is mainly we are writing to make the person to understand the safety profile of the drug. Previously, we have done this one in animals. No animals were died or no adverse reactions were happened to the animals. This is having some safety profile, and even we can use this in human beings also like that. To give the information to the reader who is reading the protocol, we have to mention these all things. Okay.
Okay.
Next, summary of the known and potential risks and benefits, if any, to human subjects. Even we have to mention about what are the benefits of this drug and what are the side effects when we are going to take this drug. What are the precautions they have to take when they are going to take in human beings. Though that information also we have to give, because in when we are conducting in animals, we may see some adverse events or some adverse reactions in the animals, that we have to mention, and we have to give the precautions also.
Okay.
When you are taking this drug, along with this drug you should not take drug interactions may happen, drug food interactions may happen, with which food it will interact, with which other drugs it will interact. Those kind of information we have to give in the protocol. Okay.
Okay.
Next, description and justification for the route of administration, through which route they have to administer the drug, what is the dosage they have to take, what is the dosage regimen, that means that dose, how many times they have to take, that is nothing but the dosage regimen, and treatment periods. Treatment periods means washout period. For example, they will give us, if we are taking paracetamol, they will ask us to take for every 6 hours, because paracetamol half life will be for 6 hours, and after 6 hours it will completely excrete from our body. That's why again one more dose we have to take after the 6 hours, as washout period will happen by 6 hours. That's why
Oh,
treatment periods also we have to mention in the protocol. Next, description of the population to be studied. So on what kind of population we are going to do this trial, like what the characters they should have. That means inclusion criteria, we'll call it, the term we'll call it as inclusion criteria. What is the inclusion criteria for the patients to enroll in the study, and what is the exclusion criteria for the patients to follow, like who are going to participate in this study. Those all things we have to mention in the description of the population to be studied. Mainly description is nothing but completely will not write the inclusion and exclusion criteria. That is another section we'll have in the protocol. And background information, just we have to write with what kind of indication the people can take this medication. Like if the people is having this kind of indication, they can take this medication. Like that, we can write in the background information in a very brief manner.
okay.
Next, reference to the literature and other information which form a base of trial. So, if you have any literature of this drug, you have some literature, that literature you have to summarize in a very short way, and you have to write such that people will have a reference. Okay, already clinical trial was conducted on this product, and now they are doing to know specifically about its efficacy or the safety. Some other additional things again these people are doing like that. It will give information to the reader about the trial. Okay. Did you understand
Okay.
this one?
Yeah, ma'am.
Next, trial objectives and purpose. Next section is objectives we will write. There should be a detailed description. So whenever we are writing the objectives, we should write in a very detailed way. What is our main objective or what is our main aim of conducting this clinical trial? And different phases of clinical trial have their specific objectives which should be described in the protocol. That for different phases I'll show. See, this is the one. Phase 1, whenever we are doing phase one study mainly focus on safety. To know the safety of the drug, phase 1 trials will be done. Okay.
Okay.
Phase 2 mainly focuses on efficacy.
Okay.
Okay. And phase 3 mainly focus on both efficacy and safety. Okay, mainly
Okay.
focus on safety and efficacy. Both they will know in phase 3, and after knowing those details only, they will they will apply for the regulatory authority approval. And
Okay.
in phase 4, we'll get approval from the regulatory authority, and we can market the drug to, we can commercialize the drug into market.
Okay.
Okay, so here what I mean to say is, in objective, we should clearly know what is a phase of our clinical trial. And for example, if it is phase 1, then our objective should be to know the safety of this product in this group of population. If our trial is phase 2, we have to mention to know the efficacy of this drug in this group of population. If it is phase 3, we have to mention to know the safety and efficacy of the drug in this group of population. Like that, we have to mention our objective based on the phase of the trial.
Okay.
Okay. Next, trial design. For this trial design, I will explain you some designs. These all are different designs that I will explain you in next slides. So next is endpoints. Endpoints, these are nothing but to attain our objective, whatever the tests or whatever the things we do, those are called as endpoints. Okay, I'll tell you with a small example. For example, now I got one patient who is like I'm doing one clinical trial. I'm doing a clinical trial in patient who is suffering with endometriosis. It is a kind of gynecological condition. So now I'm giving one drug to the patient to know whether this endometriosis is getting controlled by using my drug. So mainly here what I will do, what are the main symptoms of endometriosis, I will check. The main symptom of endometriosis is pain, menstrual pain, back pain, pelvic pain. These are all some of the main symptoms or main problems that will be faced by the endometriosis patients. So now if I'm doing a clinical trial, on which I will target now?
To relieve the symptoms, pain, and
Relieve the
everything.
symptoms, to relieve the pain to the patient. So, what is my main objective here? I'm doing the phase 3 trial. Then my main objective is to know the safety and efficacy of this particular drug in relieving pain for the endometrial patient. Okay, that is my objective. To achieve that objective, in endpoints I will mention. So in endpoints I'll mention I'm doing this study for 3 months. So in these 3 months, from first month to the third month, what is the reduction of pain in that patient? So to know that one, I'll keep one questionnaire to the patient. In that questionnaire, every time when patient is coming for the visits, I'll ask her to write, okay, how about your pain? Like, what is your rating of pain in first month? What is your rating of pain in second month? What is your rating of pain in third month? Such that I can compare by the end of the study whether the pain was reduced when compared with the first month after taking my medication, right? Like that,
Yeah.
we can compare and we can know whether it is showing efficacy or not. Coming to safety, in these 3 months, did the patient is having any adverse events? Did the patient suffered with any adverse drug reactions? Those things I can check, and I can know whether the drug is acting safely or not. Okay, so
Okay.
those things, whichever I am like whichever the assessments I am doing on the patient to achieve my objective, that assessments I'll write in endpoints. Got it?
Yeah.
So that is it. That is those are the things we'll mention in the objectives and endpoints. So as I said, you these are some of the trial designs. And coming to the trial designs, I have one picture, I think. Okay. Okay. Mainly in trials, we'll do randomization. Do you know the meaning of randomization?
Uh, Randomly allocating.
Randomly allocating the drug. Yes, correct. Give me 2 minutes. So here randomization is nothing but randomly we'll allocate the drug to the patient. For example, in my trial, first I have taken 10 patients, and in my trial, placebo will be there and active drug also will be there. And I'll tell you what is meant by placebo. Placebo is the meaning that it is a kind of a medicine. It is not a medicine, but it will also look like a tablet, but it will not contain any active ingredient.
Okay.
Okay. And in my IP, investigational product, means it will contain the active ingredient. Why we will do this kind of trial is, we cannot know the efficacy of the drug until or unless we conduct a clinical trial on the patient who is not taking the drug also. But patient should not aware that he is not taking the drug. Because if the patient aware that he is taking the drug, what he will do? By his psychological condition, some people will have some psychological conditions. For example, some old people, mainly they'll have, not only old people, so many people, they'll have some psychological condition. They will feel like they are having headache, and until we give a tablet, they will not satisfied that they are relieved from the headache.
Yes.
Yes, did
Yeah.
you seen, have you seen this scenario?
Yeah. Yeah.
Yes, because
Yes.
they'll have that psychological feeling. They'll have a feeling
Yes.
that if I take a medicine, immediately I'll be get relieved. Even though it relieved, if we take rest also for some time, our headache will be reduced. But they'll feel like if I take the tablet only, that headache will be relieved. If I take that medicine only, my condition will be get proper. Like that, they'll have some psychological feeling. That's the reason here we'll use placebo in clinical trial, because in placebo we'll not have any active ingredient. Just to satisfy the psychological condition, we are giving this placebo.
Okay.
It's a kind of dummy drug.
Yes.
Okay, so we'll take two groups in the study. That is, one group will receive the placebo and one group will receive the whatever the active medicine is there, that one. Because we can know the difference from placebo group from to active group, even only if we have one comparing kind of thing. We cannot know the efficacy of the active group if we don't have any comparison. That's the reason we'll take the placebo groups also. And so here the question will arise, to which patient we have to give placebo, to which patient we have to give active drug. That's the reason here we'll do randomization. What we will do is, for example, if we have 10 patients, generally our mindset how it will be is for the first patient we'll give drug, for the second patient we'll give placebo, again for the third patient, like that. For our easy way of convenience we'll give. But due to that, what will happen? Doctor will have some favorism to the patients, and he will do favor to the patients whom he know very well. So to whoever the patient he know very well, he'll give the drug. To whomever the patient he don't like, he'll give placebo. Like this kind of things will happen. That's the reason randomization will do in the clinical trials. Randomization means randomly. Even doctor also will not know which drug he is giving to which patient. Like that,
Okay.
we'll give some coding. First patient, code A. Second patient, code B. Third patient, again code B. Like that, we'll give some codes. Which medication we are giving to which patient will be known only to the CRO people, not to the sponsor only. Like that kind
Okay.
of blinding we will do in studies to avoid the bias or to avoid the favorism. Okay. So in
Okay.
that situation, we'll give the drugs very randomly. For example, out of 10 patients, only for the sixth patient, our second patient, our first patient, we don't know whoever we are giving the drug, to whomever we are giving placebo. Like that kind of randomization we'll do, and we'll give medication to the clinical trial group. Okay, that's why here we have different types of designs. First, as I said, randomization is nothing but giving the drug randomly to some patients. That is randomization. And another thing is blinding. As I said, we'll blind the information. We'll not let the information known to anyone, or mainly to doctor or patient in the clinical trial. That kind of blinding we will do. In blinding, we have different kinds. Like in some studies, we'll do single blind. In some studies, we'll do double blind, triple blind, like that. I'll tell you what is meant by single blind, double blind, and triple blind. In single blind, patient will not know the information of what drug he is taking, because as per our clinical trial procedures, what we have to do, we should inform to patient everything, what he is taking, why he is taking, what are the benefits, what are the risks, right? But in this blinding, single blind will not let the know, we will not give the information to patient what he is taking. Got it?
Yeah, yeah, yeah.
That is single blind. In double blind, doctor also will not know, patient will not know, and doctor also will
Doctor
not.
also.
In single
Okay.
blind, doctor will know, but patient will not know.
Okay.
Okay. In double
Yeah, yeah.
blind, doctor and patient both will not
Both
know
the
what
same.
drug they are giving. What drug they are giving, doctor will not know. What drug they are taking, patient will not know. In
Okay.
triple blind, doctor, patient, and sponsor also will not know. Only statistician will know. Whoever write the code for those drugs, that person only will know which drug is going to which patient.
Okay.
Okay? These are the different kinds of blinding we'll have in the clinical trial.
Okay.
That is about the design of the study.
Okay.
Next,
Ma'am, so what is parallel design?
parallel means parallelly, simultaneously. For example, we have two groups. In one group, we are giving placebo. In one group, we are giving active drug. Parallelly, in that group, for the first 30 days, we'll be giving placebo, and parallelly here, in the second group, we'll be giving the IP. There will be no gap. Sometimes what will happen is same patients will be participating in the study. At that time, first for the 30 days, we'll give drug A to that A set of patients, and we'll take some time gap, and then again to that same group only we'll give another drug. But here
Okay.
parallel means there will be no gap. In the first set of patients for the first 30 days we'll be giving, and in the second set of patients also for the first 30 days only we'll be giving the different drug.
Okay.
Parallelly it will happen at the same time. The drug taking will happen in two groups.
Okay.
Next, inclusion and exclusion criteria. So mainly whenever we are doing the trials, whoever the patients we are enrolling into the study, for them we have to take some considerations as inclusion, and some considerations into exclusion. So exclusion means if they are meeting those criteria, they should not participate in.
Include.
That is the meaning of exclusion. Okay. So in inclusion criteria, some of the main things which we have to consider are age of the patient and gender. Sex means gender of the patient, type and stage of disease. For example, we are doing kidney disease clinical trial. So whenever we are taking the patients for that kidney disease trial, we should take the patient who are having only the stage, only up to this stage, kidney stage. We'll have some kidney disease. We'll have stage 1, stage 2, stage 3A, 3B, 4. Like that, we'll have different stages in kidney disease. So up to stage 3 only patient should be taken for this study. Like that, we'll mention in the inclusion criteria, and those kind of patients only should be enrolled. Okay.
Yes.
And in treatment history also, we'll keep some limitations. Up, whoever having the treatment history of dialysis or transplantation, those kind of patients only should be enrolled in the study. Like that, we'll keep some limitations. So that treatment history also we have to mention whenever we are writing for some particular indication to a patient, or when we are enrolling the patient into the study, or other medical conditions. These all we have to keep in mind when we are designing inclusion criteria for the clinical trial. Okay. And some of the exclusion criteria which we have to consider are mentally retarded patients. These mentally retarded patients should not be included in the clinical trial. They should be excluded. Unconscious patients also should not be included in the clinical trial. They are the excluded patients. Pregnant women should not participate in the clinical trial. That's why they should be excluded. Patients who are unwilling to sign informed consent form, those kind of patients also we have to keep in exclusion criteria. So as per based on our study, we have to design the inclusion and exclusion criteria. But these things should be definitely mentioned in the exclusion criteria. These 4 things, whichever I said: mentally retarded patients, unconscious patients, pregnant women, and patients who are unwilling to sign the informed consent form. Okay.
Okay.
Any doubts?
No, ma'am.
Next, even in clinical trials, if the patient is participating in the clinical trial, for example, the study is for 3 months. He participated in the study up to 1 month, and later he felt, I don't want to participate in the study. I'm not feeling well after participating in the study. Like that, they'll have some feeling. So those kind of patients, even in middle of the study also, they can withdraw from the study. Okay. Here,
Okay.
subject is nothing but the patient who is participating in the trial. That one we'll call, that person who is participating in the study, we'll call that person as subject. Okay. So, subject's withdrawal criteria. So, we have to mention in the protocol about subject withdrawal criteria also. If the patient is, when and how the patient can withdraw from the study, and the type of timing and data to be collected. For example, if any of the patient is withdrawing from the study, then what type of data or what type of things we have to collect from that subject. Because simply if the patient is saying, I want to withdraw from the study, we can tell, okay, you can go. Like that, we cannot. We should ask him, what is the reason why you are withdrawing from the study? What makes you uncomfortable to withdraw from the study? Like those kind of things we have to know, because we should know the reason also when the patient is leaving from the study, right? So those all things will be mentioned in the protocol. And in case if the patient is withdrawing, whether we have to replace another patient in that place of withdrawal patient, those all things, those conditions, because these will differ from study to study. These will not remain same for every study. That's the reason these kind of things we have to mention in the protocol. Mainly, what is the procedure for the treatment of subjects? So treatment administered to the subject, whatever the treatment we are doing in the study, that treatment will give to the patient. Medication permitted before or during the trial. Procedure for monitoring. So what we'll have one section in the protocol, that is how we are going to treat the subject. So in that one, first we have to take whether our medication is permitted before or during the trial, that one we have to write, and what treatment we are going to do to the patient, that one we have to write. And after giving our treatment, what are our monitoring processes, like what follow-up we are done, or on which time periods we are doing follow-up with the patient to know the efficacy or safety of the drug. Those all things we have to mention in the protocol. Next, assessment of efficacy and safety. As I said, efficacy and safety parameters we have to mention in the endpoints. And for efficacy, we have to, as I said, when we are giving drug to the patient, how his pain is getting reduced, how his quality of life is there after taking that medication. That one, those kind of parameters we'll mention in efficacy. Safety is nothing but we'll mention whether the patient is having adverse events or adverse drug reactions after taking this drug. Because if the patient is not having any adverse events and adverse drug reactions after taking the drug, it means that the drug is very safe to the patient, right? By knowing AEs and ADRs only, we can assess whether the drug is safe to the patient or not. That's the reason. In assessment of efficacy, we have to mention the parameters which will help us to know about the efficacy of the drug, like questionnaires, lab tests, by seeing the reports of lab tests, by seeing the questionnaire, or by taking the survey from the patients. From these things, we can know whether how the drug is showing efficacy on the patient. Next up, safety is by knowing the, for example, in the patient in 1 month period, if he don't have adverse effects or adverse drug reactions, then from that we can know, okay, this drug is safe and no side effects are shown on the patient. So that's the reason we have to mention the assessments for efficacy separately, and assessments for efficacy separately, and assessments for safety
separately.
Next, statistical section. In statistical section, mainly they will mention about how many patients are enrolling into the study, and after taking the data from the, for each data, how we are going to analyze. Before going into the statistics team, after collecting data from the site, first it will go to clinical data management team only. So clinical data management, they will review each and every data of the patient, his prior medication, his laboratory reports, his con medication sheets, his assessment parameters, which will help us to know the efficacy, the AEs, ADRs, everything they will check, and they will maintain it in a proper Excel file. And that Excel file, they'll share it with the statistics department. Statistics department, they'll have some software through which they will run the data, and they'll give the analysis, statistical analysis they'll give, whether the results which we obtained are statistically significant or not. So this drug is showing good efficacy or not. Those all things we can know through the statistical section, like t-test, ANOVA. Some of the methods they will implement, and they will determine whether the drug is acting properly on the patient or not. Okay.
Okay.
Quality control and quality assurance department. So these two departments, what they will do is mainly they will check whether all the procedures are being done as per the guidelines, all the documents are as per the guidelines or not, everything is done in an ethical way or not. Those all things they will cross check, and their main duty is to maintain um, quality of the documents, mainly, like without any manipulations. For example, in academics, what we'll be doing, even though we'll not get the results, we'll manipulate the results and we'll write the results, right? But here,
Mm.
as we are doing the trial on human beings, whatever the results we are getting, the results we have to show in a proper way. Even though patient is having adverse effects, we should not show that the patient is not having adverse effects. Whatever is happening, that we have to maintain in a proper way, such that the drug, as the drug is released into the market, it should not show effect on large group of population. That's the reason we are doing clinical trials. So these all data, whichever the data we are giving is having originality or not, are we manipulating anything, are we maintaining proper documents, this all will be checked by the quality control and quality assurance departments. Mm. So that quality assurance and quality control things also, like simply we'll write what the procedure they will be doing in the study, that also we will write in the protocol. Okay.
Okay.
So these all things will present in the protocol. Even I can share you one protocol, but as I like that my company thinks, but I'll share you one protocol for your reference, but not now. By Monday, I will share one a simple protocol to you to know what we'll mention in introduction, what we'll mention in background, rational, inclusion criteria, exclusion criteria, objectives, endpoints. These all things you can know by reading one protocol. So that protocol I'll share with you on Monday. Okay.
Okay. Ma'am,
Right?
this protocol should be written by
Medical writing team.
medical writing
Hmm.
team.
So medical writing team will write the protocol. CDM team, what they will do is in protocol, endpoints will be there, right? For
Yeah.
whatever our objectives, for those objectives will have endpoints. So as per that endpoints, all the results did we reached or not, did we received or not, that one will be checked by the CDM. And as per the endpoints only, you have to extract the data.
Okay. Okay.
That I will show you. To how to extract the data, we can know from CRF. Our next class will be on CRF only, case report forms.
Okay.
That I will show you in next
Okay.
class.
Okay. Ma'am,
And
so next class will be
Monday.
Monday.
Weekends, I will not take any class. Up
Okay.
to Friday only, I will take the class.
Monday to Friday. So Monday at what time, ma'am? Like this on.
Monday, same time, 11 to 12.
Okay.
Okay. So
Okay.
again, we'll meet on Monday, and from Monday, your core CDM topics will start. That I will start from Monday. These are
Okay.
basics that you should have.
Okay. Okay. So,
Uh,
ma'am, one doubt, ma'am. What is the difference between efficacy and effectiveness?
Same thing.
Same thing.
Same thing. Effectiveness, efficacy, like short, concising the term. That's it.
Okay. So, thank you, ma'am.
Thank you. Again, we'll see on Monday.
Okay, ma'am. Bye.
Bye.